In the last video where we talked about blast results, we left off at this page. So what can you do from this page? Well, if I click on one checkbox here in the first column, look what happens to the green action bar, all the things that you can do about it. You can find out information. You can talk with Copilot about things you could do next. You could download the sequence. You could copy the rows. I do this a lot when I'm looking at the variation service. You could look at other services that you could do as downstream analyses. You can look at subsystems to see what this gene might be involved in, or pathways. You can get the sequence. You could map it to different IDs. You could create a group of it, either a group of the genome. Well, you wouldn't want to do it with one. You'd actually want to do it with all of them. And so you could create a group of these genomes or of these features, which are the genes or proteins. You could see the genomes themselves. You could open a new tab that showed you these particular genomes or those features. But we don't want to do that much. This time I want to go to the feature page, which is all the information about this protein and this particular protein in this particular genome. So I click on that F for feature, not for failing grade. It's F for feature, and it's going to load the feature tab. Generally at the top of any page in BV-BRC, it gives you a map telling you, you are here. We're at the landing page for this gene protein in this genome, which belongs to this species, this genus, and all the way up. And anything that's blue is a hyperlink, and you can click on that, and it'll take you to the landing page for that particular thing. You notice we have the information about the genome ID. Remember, we blasted the PBP2X gene that we got from GenBank against the BV-BRC database. We asked for only 10 results out of all the Streptococcus. This was one of the hits, and it was this particular genome. I mean, conceivably, because this is an essential gene that the organism can't survive against, if we could blast and record the results for as many genomes as there are of Streptococcus, we would have that many results because the organism does not survive without this. But it's this particular genome that belongs in this taxon. This is just telling us the annotation information, what the gene is, which matches here. Its start, its end, it's on the reverse strand, and the location of it. The accession number, which contig is it in this particular genome, where it starts and ends on that contig. At this point, you could view the nucleotide sequence or the protein sequence. It tells you what it does. Notice that it doesn't say PBB2X, because when these proteins were originally described, they were described in the context of antimicrobial resistance. Now people are moving toward describing them in the function that they have within the organism itself, which it is a cell division protein, FTSI. Okay, so that's what they're called now. The local family ID, the global family ID, the Go term, and when this gene on this genome was inserted into the BV-BRC database. This shows you where it is on that contig. And it's even got, if I mouse over that, it shows that there's a classifier. When we did our machine learning algorithms on them, it identified this as a tiny region that's associated with susceptibility or resistance to a particular class of antibiotics, the beta-lactam antibiotics, of which penicillin is one, or the penicillin group. So we have more information. You could get all the proteins that are assigned to this protein family, and there would be a lot. All the proteins assigned to the global protein family, and there would be even more because that passes all bacteria. You could see the proteins that are identical, the genes that are identical, and once again, the Go term and what subsystem it's involved in. Every genome that comes into BV-BRC is blatted against specialty databases, and this is hitting them. One is for antibiotic resistance, and it's showing those hits. Also for drug targets from Drug Bank and TTD. So these are the different databases that it's hitting. But wait, that's not all. There's even more on this page. You can look at the genome browser. Looking across the whole genome, it'll show you this particular gene. So let's click on that. Okay. It's popping up, and you notice that bright yellow spot? It's showing you where your gene is in the grand scheme of things, and you can zoom out and see more, or you can zoom in and in and in to where you can see the 6' translation. I can drag it around. And here is that classifier region. You can see that here that is associated from the machine learning algorithms that we run. We did the training with these data sets. You can see exactly where that is and exactly what that sequence is. You can look at the compare region view, looking at that across different genomes. This sometimes takes a while, but we're going to click on it. This is looking first at the gene and then expanding out to the neighborhood. Sort of like what BLAST does. It looks at a smaller area and then expands out. It first starts with the reference and representative genomes, and then you could choose other things that you wanted to look at on this. But just for a few points of reference, the red is my genome. And look, there's that classifier region too. And it blasts against that same genome. So it's showing the strength of the blast hit. Because this is looking across all the reference and representative genomes, it's showing it in something totally unrelated. But you can see that the red still is indicating that this is the reference gene, but it's a weaker hit. If we were to change it to all genomes, I guarantee you this would all be red for Streptococcus. Transcriptomics, we're not going to see any of that because it's not a reference genome. Interactions, those are for the reference genomes. Domains and motifs, we'll see. Yeah, generally those are reference genomes, as are protein structures. So overall, this is a feature page, and these are the things that you can see on it.